Hi everyone, my name is Tanya, and today I'll be exploring the neural and behavioral mechanisms of pain in Aplysia, the sea slug. What fascinates me about this topic is how such a simple animal can blur the line between reflex and experience. When Aplysia is harmed, it doesn't just react, it learns, remembers, and changes its behavior. So my project asks a deeper question. If even a sea slug can learn from harm, where does pain, real felt pain, truly begin? Here's a quick outline of what we'll be covering today. I'll start by introducing why studying pain in simple animals matters, then move into the neural and behavioral evidence in Aplysia. After that, we'll look at the broader pain debate, its evolutionary significance, and what this means ethically. Finally, I'll end with some key takeaways and implications. Pain is one of biology's most powerful defense systems. It protects animals by helping them avoid harm. But not every reaction to damage is truly pain. Nociception refers to the neural detection of something harmful, like when you automatically pull your hand away from a hot surface. Pain, on the other hand, is the conscious feeling of distress that comes afterwards. It's personal, emotional, and much harder to define. Understanding where nociception ends and pain begins helps us trace how complex experiences evolved. And that's exactly what my project explores. To really evaluate whether aplasia might feel pain, we first need to define what pain actually means in a biological sense. Scientists generally agree that pain involves several components beyond just detecting harm. The first is detection, the ability to sense and respond to damaging stimuli. The second is learning and memory, using experience to modify behavior after harm. Then there's motivation, when an animal prioritizes protection over things like feeding or exploring. A fourth is emotion or effect, where the response lasts beyond the moment of injury, suggesting some kind of internal state. And finally, integration, the neural processing that ties these components all together. In the next few slides, we'll test aplasia against each of these to see how far along the spectrum of pain it really falls. In vertebrates, pain is clearly linked to conscious processing. Brain regions like the cortex and amygdala help generate emotional subjective experience. But invertebrates don't have those structures, which makes things a little bit more complicated. Some, like octopuses, show surprisingly complex behavior that hint at pain-like emotions, while in others the evidence is far less clear. That's where Aplasia, the sea hare, comes in, a small marine invertebrate that it sits right in that gray zone between reflex and experience. It's simple enough to study neuron by neuron, but it's still capable of surprisingly complex learning. Let's take a closer look into that. This is Alplasia, also known as the sea hare. It's one of neuroscience's most iconic model organisms, not because it's complex, but because of how beautifully simple it is. Alplasia has around 20,000 neurons, many which are large and individually identifiable. That allows researchers to actually trace how a single neuron contributes to a specific behavior, something that's nearly impossible in most animals. One of its key defense behaviors is the gill and siphon withdrawal reflex. When it's touched or receives a mild electric shock, Alplasia automatically contracts its gill to protect itself, something we'll look into more in the future slides. What makes this reflex remarkable is that it isn't fixed. It changes with experience. With repeated stimulation, Alplasia becomes sensitized, meaning it learns to react more strongly after being harmed. The molecules that drive this process, serotonin, PK, and Krebs, are the same ones that are involved in learning and memory in humans. So even in a nervous system this simple, we can see the building blocks of pain-related learning, how organisms begin to remember and adapt to pain. Alplasia protects itself with a simple reflex. When its siphon or tail is touched, sensory neurons activate motor neurons that make its gill contract. That's the basic reflex circuit. But what happens when this circuit experiences harm? After a tail shock, a different set of neurons release serotonin onto that same sensory neuron. Serotonin strengthens the sensory to motor connection through molecular messengers like PK and Krebs. These molecules change how the synapse functions. It releases more neurotransmitter and stays active longer. With repeated shocks, the same pathways even switch on gene expression, leading to lasting structural changes. So harm doesn't just trigger a reaction. It literally rewires the reflex. And what's striking is that these same serotonin-driven mechanisms underlie memory and chronic pain in humans. In one small circuit, Aplasia shows us how experience turns reflex into memory and reveals the cellular roots of learning from pain. This short clip over here shows Alplasia exhibiting its gill withdrawal reflex. You'll see how with a gentle touch, it just causes a small contraction, but after a noxious tail shock, the withdrawal is much stronger, evidence of sensitization. So I'll let the video show it for you.
Weak stimulus to the siphon, you see a modest withdrawal of the gill. Now we're going to frighten the animal, startle it, give a noxious stimulus to the tail. That, of course, causes a contraction in its own right. This contraction lasts for seconds, but you can come back minutes later. The same siphon stimulus now produces a much more powerful withdrawal. We can now compare the two, normal and sensitized, and see how much more powerful the withdrawal is in the animal that is startled. The memory for this event.
So even in a simple animal like Alplasia, we can see its nervous system adapting through experience. That link between harm, memory, and behavior, it's exactly what connects reflex to experience. After harm, Aplasia's withdrawal reflex doesn't just recover, it stays heightened for over 24 hours, as you can see over here. This shows that the experience of harm leaves a long-term trace, mediated by the serotonin-driven pathways we've already seen, PK and CREB. This response is also context-specific. If the animal was tested in the same environment where it was shocked, it reacts more strongly. That kind of learning, linking place and pain, is the same mechanism that supports aversive memory in invertebrates. This satisfies the second criterion of pain, learning and memory, the nervous system's ability to hold on to harm. Now, Alplasia doesn't just learn from harm, its motivation changes too. Injured animals feed less, explore less, and remain partially contracted long after the initial shock. These behaviors show a motivational shift. Energy is redirected from normal activity towards protection, a hallmark of pain in higher animals. In vertebrates, this state of pain is mediated by affective brain regions, like I already discussed, like the amygdala. In Alplasia, it's achieved through prolonged sensitization in peripheral circuits, a simpler but functionally similar system. This hits the third criterion of pain, motivation, the internal reorganization of priorities after harm. Even after the injury heals, Alplasia remains sensitized. Its defensive reflex stays amplified for days. Studies by other researchers show that this long-term hyperreactivity persists even in the absence of ongoing harm. Functionally, that persistence acts like a primitive emotional state, a lingering readiness to defend, similar to anxiety or discomfort in vertebrates. This fulfills the fourth criterion of pain, a lasting affective state that outlives the initial stimulus. It's what makes pain more than a reaction. It makes it an experience. So, after looking at all of the evidence, it's time to come back to our central question. Can Alplasia feel pain, or is it just reacting to harm? If we define pain as a set of biological functions, detection, learning, memory, motivation, and effect, then Alplasia clearly meets nearly every one of these components. It detects harm through specialized sensory neurons, it learns from experience through serotonin, PK, and CREB, and it changes its priorities after injury, feeding less and defending more. It even shows a lasting defensive state, a kind of primitive distress that persists long after the injury ends. What it lacks is the integrated consciousness that gives rise to what we call subjective suffering. But if pain evolved as a way for nervous systems to remember and avoid harm, then Alplasia expresses an ancestral form of pain, a biological forerunner to the emotional experience that we feel. So rather than saying it doesn't feel pain, it's more accurate to say Alplasia shows us where pain begins, when a nervous system starts to connect harm with memory, emotion, and self-preservation. From an evolutionary perspective, pain-like systems didn't evolve to make life harder. They evolved to make life last longer. The earliest animals could react to harm, but species like Alplasia added something new, the ability to remember harm and adapt their behavior. That shift from reacting to learning is one of the most important transitions in the history of nervous systems. As evolution continued, those same protective functions diversified. In octopuses, pain became tied to complex decision-making. In humans, it became emotional and self-reflective. But the purpose has never changed. Pain evolved as the body's biological memory of danger, a way to prevent harm before it happens. So when we study aplasia, we're not just looking at a primitive reflex, we're looking at the origin of feeling itself. The discoveries made in aplasia didn't just reveal how animals learn, they changed how we understand pain itself. In humans, chronic pain isn't simply a prolonged injury, it's a form of maladaptive learning. The brain keeps replaying the signal long after the danger is gone. Pain becomes a memory refusing to fade. Alplasia gives us the simplest version of that process, when harm leaves a molecular mark that changes how neurons communicate. But understanding how alplasia rewires circuits after harm, scientists can better grasp how human pain pathways become oversensitized, and more importantly, how they might be reversed. So, this tiny animal doesn't just model pain, it helps us understand why suffering persists and how healing might begin at the cellular level. But beyond medicine, this research forces us to ask a deeper, more human question. If we accept alplasia can feel if we accept that alplasia can remember harm, that it changes its entire way of being after injury, we have to ask a harder question. Where does awareness begin? Does it require language, a brain, emotion, or is awareness something much more simpler? The moment a nervous system learns to remember harm. If even the simplest nervous systems can remember pain, then maybe compassion isn't something uniquely human. Maybe it's an ancient inheritance written into the biology of life itself. Maybe pain isn't what separates us from other animals. Maybe it's what connects us. So let's bring all these ideas together. To summarize, we began by asking whether simple animals can feel pain. Alplasia has shown the line between reacting and experiencing isn't sharp at all. Its neuroplasticity, serotonin, PKA, CREB shows how injuries can leave a memory trace. Behaviorally, it learns to avoid harm and change its motivation, revealing primitive awareness. Evolutionary, it connects us to every organism that's ever learned from danger. Ethically, it challenges us to see compassion not as a human trait, but as a biological one. In the end, alplasia demonstrates that pain isn't just a signal, it's an adaptation, a memory, and perhaps the first whisper of consciousness. Thank you for listening. Let me know if you have any questions.